Hello, everyone. My name is Madeline Greth, and I'm going to be presenting on glycolysis, gluconeogenesis, and the role of aldolase. So first, let's go over what glycolysis is. So glycolysis is a metabolic pathway and an anaerobic energy source. This can also be used for aerobic energy source. It's the first step in cellular respiration. It does take place in the cytosol. It is the oxidation of glucose molecules. Glucose is the most crucial organic fuel molecule for plants, microbes, and animals. Most cells do prefer glucose. Of course, there are always some exceptions. So during this process, 2 ATP molecules are consumed. This is what's called the investment phase. The cell is investing these ATP molecules to be able to get to the payoff phase. In the payoff phase, this is when 4 ATP are produced, 2 NADH are produced, and 2 pyruvates are produced. And then these pyruvates are later used for other steps. We'll dive deeper into this process in the next slide. Like I said before, glycolysis is occurring in both the aerobic and the anaerobic states. In aerobic conditions, the pyruvate will enter the citric acid cycle. It will undergo oxidative phosphorylation, which leads to the net production of 32 ATP molecules. In anaerobic conditions, however, the pyruvate converts to lactate through anaerobic glycolysis. All right, so the steps of glycolysis broken down. So the first step is the conversion of glucose to glucose 6-phosphate by the enzyme hexokinase or glucokinase in the liver and the pancreas. This does use an ATP and a phosphate group to do this first step. Second is glucose 6-phosphate, which is converted to fructose 6-phosphate, an isomer, and it's converted by phosphohexose isomerase. Next, the fructose 6-phosphate is converted to fructose 1,6-biphosphate. This is done by phosphofructose kinase, and this is where that second ATP molecule is being utilized. And then fructose 1,6-biphosphate is converted to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, also known as DHAP. And these are converted by fructose biphosphate aldolase. The DHAP is then converted into glyceraldehyde 3-phosphate as well by the triose phosphate isomerase. And each of these molecules now follows the same pathway in the payoff phase. It's just done twice, once with each of those molecules. All right, so that was the investment phase. Next is the payoff phase. So the steps here are the dihydroxyacetone phosphates are then converted to glyceraldehyde 3-phosphates. The glyceraldehyde 3-phosphates are oxidized to 1,3-biphosphoglycerate. And during this process, you're reducing the NAD plus to NADH in a proton. Then 1,3-biphosphoglycerate is converted to 3-phosphoglycerate with the help of phosphoglycerate kinase. And here is when we're creating our first ATP molecule. Now, it's doing this twice. Because again, I said it's going through that payoff phase twice with each of those compounds. So you're getting 2 NADH in the previous step, and now in this step, you'll be getting 2 ATP from that. Next, the 3-phosphoglycerate is converted to 2-phosphoglycerate by phosphoglycerate mutase. 2-phosphoglycerate is converted to phosphoenolpyruvate. This is done by releasing an H2O molecule with the help of enolase. And then pyruvate kinase then facilitates the loss of a phosphate group to create another molecule of ATP, and then converts phosphoenolpyruvate enolpyruvate to pyruvate, which we talked about as that end product from glycolysis, which would then go into either lactic acid cycle or citric acid cycle. All right, so this is just another way to kind of break down glycolysis. Here you have the 10 steps listed. On the left, you have all of the enzymes in the order that they're used. On the right, you can kind of see what's being used and what's being produced. So in those first few steps, that investment, you use ATP and a phosphate, and then you use another ATP to use a total of 2. And then in the last stages, you're reducing NAD to get NADH twice, and then you're producing ATP twice, but then for both of those molecules. So total of 4 ATP that you're creating. All right, so the function of pyruvate. So pyruvate continues to the citric acid cycle. This can be used as an emergency backup for cellular respiration. So this can be seen in that lactic acid fermentation, which I'll get to in just a second. It's also used for acetyl-CoA synthesis and L-malate synthesis, and a precursor to other molecules. So pyruvate, very important compound used in many different processes within the body. So emergency backup for cellular respiration, what we were talking about, this is that lactic acid fermentation. So cells with no mitochondria are going to be using lactic acid fermentation all the time. These are things such as the lens of the eye and erythrocytes. The lens of the eye, you can't have mitochondria or else it would actually cause light scattering. And then skeletal muscles. So this is that emergency backup for cellular respiration. This is when you're doing heavy exercise, there's not enough oxygen for the demand of the muscles. So the muscles start to undergo anaerobic and aerobic respiration. It's what makes you sore, that lactic acid buildup. Just another way for your body to kind of get that ATP molecule. Not as efficient, but it'll have to do. All right, regulators. So we have the GLUT transporters. Regulation of these transporters, they transport the glucose from outside the cell into it. Cells can increase the number of transporters that it needs depending on the energy needs of the cell. There are 5 types, and then they're all present in different kind of tissues. They're called GLUT1, GLUT2, GLUT3, GLUT4, GLUT5. And then the breakdown storage of glycogen is another regulator. When glucose levels are too high in the cell plasma, the cell can store the glucose as glycogen that can be used in times of low glucose. So when there is low glucose, there are 2 enzymes that control the breakdown of glycogen: glycogen phosphorylase and glycogen synthase. The enzymes are regulated through feedback loops. All right, and then you have the opposite of glycolysis, which is gluconeogenesis. What this is used for is this is a method that synthesizes glucose from non-carbohydrate precursors. So it's going to be converting, we've been exploring, kind of how the body uses glucose, and glucose remains the almost universal fuel and building block in most of the modern organisms. Glucose is necessary for continued function, with some cells relying completely on glucose as their energy source. These are things such as the brain, nervous system, erythrocytes, embryonic tissue, areas of the body where you really don't want to be running out of glucose. So when your glycogen stores are depleted, your body needs a different way to be able to get glucose. And the way that it does this is through gluconeogenesis. So it's converting pyruvate and other 3 or 4 carbon sugars into glucose. So this is done mainly in the liver, but also could be taking place in the renal cortex and epithelial cells. The Cori cycle, so this is lactase. The lactate is the lactate that is produced during the vigorous exercise will then return to the liver and will be converted to glucose, which then moves back to the skeletal muscles and then is converted back to glycogen to be stored. It's very similar to the reverse of glycolysis, though there are 3 reactions that cannot be reversed. And we'll go into those, and we call these the bypass reactions. So the steps of gluconeogenesis. So the first step we have is pyruvate to oxaloacetate, which then to phosphoenolpyruvate. So this is our first bypass step. So pyruvate to oxaloacetate to phospho, step 1. So it's 2 exergonic reactions that are happening. These reactions are taking place in both the mitochondria and in the cytosol. The pyruvate is either transported into the mitochondria from the cytosol or is generated from alanine within the mitochondria. The enzyme pyruvate carboxylase with the coenzyme biotin uses 1 ATP to convert the bicarbonate and the pyruvate to oxaloacetate. And that's what you see here on the left. So that was step 1. And then step 2 in this reaction, oxaloacetate is converted to the phosphoenolpyruvate by PEP carboxykinase. During this phase, CO2 is lost and a GTP is used. All right, so then the next step of gluconeogenesis, we have the phosphoenolpyruvate being converted to 2-phosphoglycerate by enolase, the 2-phosphoglycerate to 3-phosphoglycerate by phosphoglycerate mutase, 3-phosphoglycerate to 1,2-biphosphoglycerate by the phosphoglycerate kinase, 1,2-biphosphoglycerate to glyceraldehyde 3-phosphate by the glyceraldehyde phosphate dehydrogenase. Now, these are all just the reverse of glycolysis. Nothing is different from these reactions. And then we have the glyceraldehyde 3-phosphate is being converted to that DHAP by the triose phosphate isomerase. 1 DHAP and then 1 glyceraldehyde 3-phosphate are then converted to the fructose 1,6-biphosphate by aldolase. Now, the next step is unique to gluconeogenesis. This is when the fructose 1,6-biphosphate is converted to fructose 6-phosphate by fructose 1,6-biphosphatase. And then fructose 6-phosphate to glucose 6-phosphate by phosphohexose isomerase. And then the last step is the last bypass step. This is when glucose 6-phosphate is converted to glucose. This is done by dephosphorylation of the glucose 6-phosphate by glucose 6-phosphate, a magnesium activated enzyme. It does not require the synthesis of ATP, which would be the reverse of the first step of glycolysis. This would just, it would take too much to be able to reverse it. So that's why this glucose 6-phosphatase is used instead. All right, so again, this is just a different way to kind of look at gluconeogenesis. This has the steps laid out in the enzymes that are used for each of those steps. Again, it's a reverse of glycolysis with 3 of those enzymes being different than in glycolysis. All right, so then the sum of the biosynthetic reactions leading from pyruvate to free blood glucose, you'll see here, you have to start with the 2-pyruvate. You have to add the 4 ATP, 2 GTP, 2 NADH, 2 hydrogens, 4 water molecules, and then that gives you glucose, 4 ATP, 2 GDP, 6 phosphates, and 2 NAD+. All right, so one of the enzymes that I had talked about during that was aldolase, which is also known as fructose biphosphate aldolase. So this is an enzyme. It catalyzes step 4 in glycolysis and step 7 in gluconeogenesis. This uses a shift-based group of compounds that are characterized by the presence of a double bond link between a carbon and a nitrogen atom. That double bond allows for the movement of electrons. All right, so here you kind of have the steps of how aldolase works. So in the first step, you have an open ring fructose enter the enzyme, and it binds to the lysine with the oxygen taking hydrogen from the glutamic acid. Next, the arginine takes a hydrogen from the lysine. It forms a double bond between the lysine and the carbon, and it kicks off the oxygen in the form of water. Then lysine will take the hydrogen from the oxygen, forming a carbon-oxygen double bond, which breaks the carbon-carbon bond, forming a new carbon-carbon double bond and breaking off one of the carbon-nitrogen bonds. At this point, the first product, which is that glyceraldehyde 3-phosphate, is formed, and it will leave the enzyme. And then in the fourth step, the lysine reforms the nitrogen carbon double bond. It breaks the carbon carbon double bond and will take the hydrogen from arginine. Steps 5 and 6 are the shift base reversal. In the fifth step, water is added, which breaks one of the nitrogen carbon double bonds. It adds a carbon oxygen bond. And then in the sixth step, oxygen forms a double bond with the carbon. The nitrogen carbon bond is broken and the hydrogen is taken away from the oxygen. And the second product, which is that DHAP, is then released. So aldolase, what is aldolase? So aldolase is a tetramer. It consists of 8 alternating alpha helices and parallel B strands. And there are 3 forms of the aldolase A that are found in humans. There are 3 forms of aldolase that are found in humans. Aldolase A is found mainly in the muscles. Aldolase B is found mostly in the liver and the kidneys. And aldolase C is found in the brain. All right, so aldolase A. So aldolase A is a glycolytic enzyme. It catalyzes the cleavage of fructose 1,6-biphosphate and glycolysis. And its turnover number is actually 50-fold higher towards the glycolytic substrate fructose 1,6-biphosphate than aldolase B, and it is at least 5-fold higher than that of C, which this makes sense. This is meaning that this one is much more likely to catalyze that reaction, which is why it's found in muscle tissue. It's very much in glycolysis. It's breaking down that glucose to be used for energy. You need that high turnover rate to be able to keep up with the demands of the muscles. Aldolase B is used for fructose metabolism and gluconeogenesis mostly. So the turnover number of the B isosome towards fructose metabolism is 2 to 5-fold higher than the turnover numbers of aldolase A and C. And aldolase B towards the 2 trioses, G3P and DHB, makes it kinetically well suited for its role. So aldolase B is more oftentimes seen in the gluconeogenesis when you're trying to create more glucose, which makes sense because it is seen more in the liver and the kidneys, which is where we talked about is where gluconeogenesis is taking place in the body. And then we have aldolase C. So this is found in tissues of constant metabolic activity. We said this was found mostly in the brain. So this kind of isn't better at gluconeogenesis or glycolysis compared to the other 2. It's kind of right in the middle. It's an intermediate. So it's just able to kind of keep up with the energy demands of something that can't really withstand being deprived of energy, such as your brain. So it's able to use it correctly, but also make it if it needs to. All right, so regulation of this aldolase C. So there are a few different factors that regulate. You have HNF1, HNF4, C/EBP, DBP. The tissue-specific transcription of the aldolase B gene is governed by a short promoter, which is stimulated by a intronic enhancer. So both the aldolase B gene promoter and enhancer fragments contain HNF1 and HNF4 binding sites. In addition to this, a binding site for the CEBP is present in the promoter. So the promoter activity was upregulated by the hepatocyte nuclear factor 1, which is the HNF1, and the CAT enhancer binding protein, which is the CEBP. But it's repressed by hepatocyte nuclear factor 3, which is HNF3. So HNF1 is tied more closely to the regulation of aldose B in the kidneys. Disruption of the ability of this HNF1 to bind will reduce the aldolase that's seen in the kidneys, but it has little effect in the liver. It was also noted that both HNF1 receptors, so there are two, and both of them are needed in the kidneys for activation. So the CEBP is more closely tied to the activation of aldolase in the liver, but it does need co-activation from one of the HFN1 to be able to stimulate that aldolase production. Hormone regulation of the aldolase B gene reflects its dual role in both glycolysis and gluconeogenesis. Glucagon and CAMP are involved in the hypoglycemic and stress conditions in which rapid increase in glucose production is essential. This requires, in particular, transient increase in aldolase B gene transcription, leading to an increase in mRNA and enzyme abundance. However, this gene must also be stimulated to allow for the proper adaptation to a shift from carbohydrate poor to a carbohydrate rich diet. So, as you can see, the process of glycolysis, the process of gluconeogenesis is extremely important for the function of your body. And there are multiple different enzymes that really catalyze these reactions and allow them all to run smoothly. We just talked about aldolase today, which is just one of these enzymes. And then kind of how, it's not just about aldolase being an enzyme, but that there's specific kinds of aldolase that are present in the different types of your tissues and your different organs, and being able to adapt to that, to have an aldolase that's better at glycolysis for those areas of your body that really need to be able to burn sugar, or aldolase being better at gluconeogenesis for those areas of your body that really need to be creating glucose for the rest of the body, to either be able to use immediately or to store as glycogen, to be able to have these glycogen stores for later. So, being able to review this and know exactly how the body's working is just a very important part of knowing why everything is working the way it is and how it's working, and then can really lead to, when things go wrong, how to kind of help it and to treat these disorders. And then my last slide are my references. So thank you very much.